for those of you who did not attend yesterday, you should be filling out one of these little cards for me. That's the information that goes on the card, your family name, your given name, why you want to be a doctor, where you're going to practice medicine, and what you are interested in. So I went through these cards last night, and so I have a couple of insights to share with you that I thought you might or might not find interesting. I had a number of people that said that they were interested in surgery. Last year I did not have a soul tell me they were interested in surgery. This brings up several very interesting things that it tells me about you. I may be wrong, but I'll tell you what my assumptions are. There are 3 kinds of learning styles. I don't know if anybody has ever mentioned this to you before, but there are basically visual learners, audio learners, and kinetic learners, okay? Obviously, visual learners are the people that read and comprehend a lot of what they read. They see pictures and they comprehend a lot of what they see. Audio learners are the people that remember a lot of what they hear. If you're one of those people who feels like you've just got to come to class because you've got to hear what the teacher says, that probably means that you're an audio learner. You probably remember less about what you read and more about what you hear. Kinetic learners are people who learn when they are moving, okay? Sometimes this causes a problem when you are like in elementary school because it's hard to learn if you're not moving and the teacher doesn't want you to move. These are the people that frequently pace back and forth when they are studying, okay? So they involve their whole body when they are learning. So people oftentimes who want to be surgeons are people who are kinetic learners, because they learn with their whole body, okay? That's not always the case, but oftentimes it means that they are doers. They always tell me, I don't like physiology because we don't do anything, okay? They're the people who like anatomy better, especially if they're dissecting, okay? There are experiments you can do in physiology. Unfortunately, we don't get to do them in this class, but if you take an experimental physiology class later, you'll get to do some experiments in physiology. But that's what it tells me if you said you liked surgeries, that probably you are a doer and that you like to be able to move and use your hands. This also brings up another part of medicine, which is called muscle memory. Anybody ever heard of muscle memory? Okay. So muscle memory is, for example, when you took the piano when you were a kid and you had to go do the recital, and they kept telling you to practice and practice and practice so that no matter how afraid you were, your hands would know what to do, okay? That is muscle memory, okay? That applies obviously to surgery in the sense that so many of the procedures you'll do while they're on a different person, and they may not be exactly the same every time, are going to require you to have the muscle memory to know where you want to make the cuts, how deep you want to cut, how far you want to go, how much the skin is going to stretch. And so you will have a lot of muscle memory in your hands for the procedures you're going to do. I had somebody that said that they were interested in orthopedics. Orthopedics has to do with bones. In America, we say this is the last bastion for the men's club. The reason I say that is because you have to be incredibly strong to do orthopedics. So, whether you are a male or female, if you are interested in orthopedics, you need to work on your upper body strength. Because I am telling you, when you have to wrestle somebody's femur together, you may be standing on the top of the OR table, because I have seen it done, and you are going to be using every muscle you have in your body, in your back, and your arms to try and align that person's leg. Now, it's not done very often anymore, but obviously if you have to use a saw and cut off somebody's leg, you also have to have a lot of muscles to do that. So that's something that, it's one of the few parts of medicine I can say actually has a physical requirement, okay? A number of people told me that they're interested in neurology, psychology, or neuroanatomy, okay? So neuroanatomy is a class that you have here, so you'll learn a lot about the sections of the brain. I personally think that it's really fascinating. My master's degree was in a neurophysiology lab where we studied memory and learning, okay? A lot of those things are actually part of your physical brain. I think we think of them as being part of our mind and just the things that we think, but they are indeed a part of your brain. There are a lot of behaviors that are directly connected to brain activity. So although that's really not a part of this class per se, when we're talking about nerves, when we're talking about neurotransmitters, I'll try to make sure that I give you as much clinical information as I can in terms of how that relates to a person's behavior or how that relates to what's going on inside the person's brain, okay? And the last one I had on here was I had a number of people who told me that they were, that they were interested in gynecology. Now, I didn't actually happen to notice if these were male or female students who said they were interested in gynecology. And I didn't know whether that meant that you were interested in the baby part of it and the development of babies and helping with childbirth, or if it meant you were interested in the other components that would have to do more with, say maybe surgery or cervical cancers or the hormones that are involved with gynecology. Unfortunately, we don't cover reproduction in this class. I voted for it, but they didn't want to give me any hours to teach reproduction. I'm assuming that means you get it someplace else, but again, when we start talking about hormones, I'll try to make sure that we talk about those components that would be of interest to people that are going into gynecology, okay? So that's kind of how I will try to focus the class on the things that it seems like you're interested in. And if I didn't mention something that you wrote on your card, you can certainly let me know in person or by email if there's something you'd like me to try and include in the class, okay? So, first of all, if you didn't come yesterday, I need you to fill out the card with this information, okay? And I'm going to take this down here in just a second so that we can go on with our lecture today. I also brought with me my USB port. So I will plug this in after class, and that means that 4 people can come stick their USB in here. It'll make it easier for everybody. Plus, we don't have to do it down at the bottom of the computer, so it won't be on the ground. Okay? Okay, let's get this all down. Okay, so today we are going to have an attendance quiz, but we're going to wait just a few minutes so that the people that went to prayers will be here. We'll give them about another 10 minutes or so to be here. So we'll go ahead and start talking about the topic today, which is membrane receptors, second messengers, and signal transduction. So please raise the hand if you've been introduced to this topic before. Anybody been introduced to cell membrane signal transduction before? Okay. 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 This is just a tiny, tiny, tiny piece of what is going on inside of a cell. This is where the forefront of medicine and scientific discovery in the medical sciences is occurring. This is also where the areas of biomedical science are merging together. It used to be at one time that if you were in pharmacology, you had this distant, removed area. If you were in cell biology or anatomy, you were over on this side. You hardly ever talked to each other. You didn't have much to do with each other. Every area was very distinct, and they had kind of their own outline of what they should study. And then cell signaling came along. Basically in the 1980s is when it started. And what happened was everybody realized they were much closer together than they thought. So that was when the pharmacology people started to realize that the medicines that they were trying to invent, we were actually involved with cell signaling. This is when all the biochemists got very excited because enzyme activity and protein activity became huge. This is where the physiologists got really excited because now there were mechanisms to explain the functions that were going on. And so all of a sudden, everything kind of got mushed together. And one of the things that's happened is that in many places in the world, you now get a PhD in biomedical sciences. You don't get a PhD in any specific, you can specialize, but you're not going to be in a specific physiology department or biochemistry department or molecular biology department because of the fact that they're now so closely related. So in this picture, it has a number of plasma membrane receptors up there at the top. And each one of them has a different job, and it has a different ligand or ligand, depending on where you come from. And each one of them has its own set of proteins and enzymes. Okay, so an enzyme is a protein with a job. Each one of these is going to have a job, sending on a message to here, sending on a message to here, sending on a message to here, and finally going into the nucleus and having a final action. So this is what we call a signal cascade, okay? All of these are pretty much proteins of one sort or another. Whoever discovers it gets to name it, okay? So usually each one of these little acronyms actually stands for something, but sometimes we use them so rarely we totally forget what they really stood for. But if you go look it up in Google, you'll find it. pKa means protein kinase A. Okay, MAP means, or I think MAP is on there someplace, means mitogen activated protein. Okay, so depending on who you were. Depends on whether you were feeling like you were being silly or not. Depends on how you got to name those proteins. So, a couple of the ones that are on here we're going to study in detail. Oops. There is a GS, a GQ, and a GI listed up there. If you look up here at the top where it says G coupled, there's a teeny little S over there, and I think this one says the GI is over there. Okay, S is stimulatory, I is inhibitory. So they work in opposites to each other. The Q kind of does its own thing. It has its whole own process. This, I think it's this one, receptor tyrosine kinase, RTK, is very important because it's very much involved in cell division. And it's very involved in cancer issues, okay? So we're going to kind of talk about some of these, and we'll talk about what their ligands are and what they are responsible for as we go through today. That's kind of what we're going to be doing. I got a couple little movies for you to reinforce how we're going to learn all these things. I'm not expecting you to learn every single one of those names. But it's really important that you understand the primary ones, and that's what we'll talk about today. Okay. So first of all, let's start with the membrane receptor. The membrane has to get a signal. That signal can come from a variety of sources. It can come from endocrine, okay? That means a hormone. In your stomach, there's a whole bunch of hormones that tell your body what kinds of things to secrete so that it can do the digestion, okay? It can be neurocrine, okay? So when we're talking about neurotransmitters, when we're talking about inhibition, when we're talking about stimulation, those are neurotransmitters, and they're going to be binding to some kind of a receptor, okay? Paracrine means that it is close to the original site. So this would be a cell that is sending something off to a close tissue, okay? May not be exactly the same kind of a cell, but it's going to be a close tissue that's close by. An autocrine means that it's a cell that will actually bring the ligand back to its own receptors. So it'll be secreted out of the cell and then back on its own receptor. So that's autocrine, okay? We also have a number of receptors that respond to the things that you would think of when you think of the five senses. So that means they will respond to light. Okay, this is how your eyes are telling what's going on. They will respond to stretch, both in your muscles and also pressure when you're touching something with your hands or you step on something, okay? And the last one is electrical. And when we talk about the voltage gated channels and stuff, you'll be able to understand where the electrical part comes into, okay? Okay. So in general, whenever we talk about a cell signaling cascade, these are going to be the basic components. First, you're going to have some kind of a ligand, and it is going to fit into a receptor. And this is like a lock and a key. So the receptor is going to be made to fit an exact copy of what it wants, okay? Now, there are times when the exact copy isn't what you get, okay? And we'll talk about that in just a minute. Next, we have the signaling cascade that's on the inside. So this is this one's outside the cell. That's the transmembrane through the cell. Now we're inside the cell. And this is the intracellular signaling pathway. Okay? So when we talk about second messengers, we're talking about this piece. They didn't receive the message, but they're passing on the message. So if I take a soccer ball and I give it to the first person on this side, and that person passes the soccer ball all the way down to the end, then we have had a second messenger system, okay? Because holding the soccer ball makes that person active. They have to actually turn and give it to the next person. The same way that getting the message from the previous from the receptor is going to make it active. So then it becomes active and it passes the message on to the next one and then on to the next one. Okay? This means that there isn't a necessity for anything to go very far in the cell. It's going to run into the next messenger before it has to go very far. In the end, we have a wide variety of things that can happen. Okay? And those are the target proteins. And depending on what we're hoping for, it may be that it is going to put a new receptor into the membrane. It may be that it's going to increase the metabolism in the cell. To increase the metabolism in the cell means that the cell's going to make more ATP. Okay? It may be that it's going to regulate a gene. If you think about it, there are a lot of genes in your body that really actually only get turned on during development. Okay? And once you are fully grown, they're never going to be turned on again. There's absolutely no need for the genes that make a kidney to make you a new one. You already got your two and you don't need any more. Okay, so they're done and they're not going to come back. There's a lot of cytoskeletal proteins that will help the cell divide, and they will also help the structure of the cell. Okay, and then the last one is the cell cycle proteins. Our cells all divide because they want to replace themselves when they get old and worn out. But if they divide too much, then that's when we have cancer, okay? So you can see how all those different things, which are mostly controlled inside of the nucleus, take place, okay? Okay, this is everything I just told you so that when you review your notes and you're looking at this PowerPoint, you got it all in writing, okay? So that's the basic idea of how the whole thing comes together. Okay, this is a very important slide because it tells you 4 things about cell signaling that are very important to remember. Number 1 is the specificity. Not every cell has every receptor. They only have the receptors that they need. It takes a lot of energy for your body to make protein. So it's not going to make anything it doesn't need. Okay? So the cells in one part of your body are not doing the same thing as the cells in another part of the body. They only have the receptors that they need. Okay? So that's one important point. The next one is amplification. So just like a pyramid scheme, you're going to start with that one receptor and that one signal. And then it is going to go down and it is going to multiply, multiply, multiply. Okay? So it is going to run into a number of second messengers, and each one of them will become bigger and bigger. So you don't have to have a large message at the beginning in order to get a lot of activity by the time you get to the nucleus, okay? So it's not going to be just one adenylase cyclase or one second messenger that gets the message. There'll be many, many that get the message, okay? All depends on who's hanging around in that same location. The other thing is that the receptors will desensitize. So in other words, they will turn themselves off, okay? There's several mechanisms that will make this happen, but once they've done their job, they're done, okay? So once they've done their job, they may be endocytosed into the cell and dissolved, or they may be removed from the cell and dissolved, but they're not going to just stay up there and continue forever to make whatever protein it is that they started with, okay? And the last thing is the integration. Okay, so imagine that you are riding in a taxi, and imagine that the taxi driver has one foot on the brake and one foot on the gas. Or if you drive a scooter, if you have your own scooter, just imagine that you've got gas and brake going at the same time. So what happens when you have the gas and the brake going at the same time? So let's say this is going to be my gas, and this is going to be my brake. Oops. Okay, so if you have gas all by itself, what happens? Do you go fast or slow? Assume you hit it all the way to the ground, okay? Just assume that you full throttle, okay? If you only have gas and nothing else, it's going to be as speedy as you want, right? If you have the brake also engaged at the same time, no matter how hard you push that gas, it's going to counter it, and you're only going to go kind of slowish, right? You might even kind of jerk along, okay? Now, what if you have no gas and you just have brake? You're not going anywhere. Okay. Your entire body, head to toe, is set up so that the gas and the brake are going at the same time, which means that all the time you are somewhere in the middle, okay? You're partly going and you're partly stopping, okay? You're set in the middle. And that means there isn't any time delay in your body between stopping, moving the foot from the gas to the brake, okay? There's no time delay. If you happen to have less gas, the brake will have more influence. If you have less brake, the gas will have more influence. But you're always kind of in there in the middle with both of them working at the same time, okay? So we're going to talk about switches, microbiology switches, and we're going to talk about things getting turned on and off. But that is just when we do that, we're just talking about one single protein. In the big picture, everything has an opposite and there's a balance, okay? So that's what keeps us in the middle, that's what keeps us in the normal range, that's what keeps us in homeostasis, okay? And the example on here on the slide shows that you've got an increased voltage and a decreased voltage, and they're going to work together. So wherever your response is, it's going to be a combination of the two, okay? Okay, some words that if you took a biochemistry class, you probably already have, but in case you didn't, just a refresher. A kinase is a protein that's an enzyme, and it's going to phosphorylate, okay? So when we talk about phosphates, what we're usually talking about is a structure that looks like this, okay? If you're talking about ATP, you're talking about 3 of these strung together, okay? Triphosphate, right? I always used to put it in my notes just as 3PO. That was triphosphate for me. Okay. A serine, threonine, tyrosine amino acid residues are the ones that are going to get phosphorylated, okay? So those are the amino acids that get phosphorylated by the kinases. A phosphatase is the opposite. The phosphatase is going to remove the phosphate So there's your balance between the kinase and the phosphatase, okay? If you're doing an experiment and you want to measure phosphorylation in a protein, the first thing you do is you add a anti-phosphatase so that they won't come unattached. Otherwise, you won't get a very good, it'll happen naturally and they'll come unattached and you won't get a good experiment. Okay, second messengers. Increasing or decreasing molecules, that's what we were talking about before. This is some of the names of them on here. Cyclic AMP, cyclic GMP, calcium, and acetyl triphosphate, IP3. And diacylglycerides, those are the ones that we're going to talk about the most today, okay? And they have the activity of getting phosphorylated by the kinases, okay? Or phosphorylating the kinases, I should say. Oops. Okay. So the first example we're going to talk about is those G-coupled protein signaling devices that we saw on that first page. And these do have an on and off switch, okay? It has an activated on and off switch depending on whether or not it has GTP or GDP that's on it, okay? That's the G protein part. That's why it's called G, okay, from the GTP, okay? Okay. There are now 800 genes that make for the GTP or the G-coupled protein receptors. This has gone up and up and up. If you have a book that was written a few years ago, it's going to tell you 300. Or maybe if it's really old, it's going to tell you 100. Okay? In terms of your books, the cell signaling is the part that has changed the most. Okay? So remember that if your book is even a few years old, that it's probably the information is not quite as up to date as it would have if you looked on the internet. Okay? Right now, we know that that's 4% of the human genome. So these signaling proteins and all the varieties of signaling proteins are a huge part of your body about what's going on. The other thing, as far as being an MD, you want to see this part, 40% of the current medicines are actually working on these G protein coupled receptors. Okay? So many of the modern medicines are right here. And we'll actually talk about some of those later. Also on this side, you'll see I've got this list of the ligands that will actually bind to these kinds of receptors. Like I said, each cell is going to have its own, the ones that it makes for itself. Whoops. Light. These are the cells that are going to be in your eyes. They're going to make it so that you can see. Odors. There's thousands of these that are connected to odors and to pheromones, okay, which also is also an odor, okay? So if you look up here at the top, I think that one says olfactory, which is going to be all the ones that have to do with odors. So yes, it is possible to get new smells, and yes, it is possible for you to smell things that you've never smelled before. It also is where we have the neurotransmitters. It says proteins and peptides. Oops, sorry. Peptides are simply strings of amino acids. They're very small. Usually a protein's like 300 amino acids or more, but a peptide can be just a string of like 3 or 5 or 8 amino acids, and yet it's big enough to do a job. So, okay. Who's seen a picture that looks like this before? Anybody ever seen one of these kinds of pictures? Okay. Let me tell you what you're looking at here. First of all, you're looking at amino acids. Each one of these little circles, and they're not actually circle-shaped, by the way, but each one of these little circles is representing an amino acid that's part of this protein. When you get into what they call the transmembrane domains, which is the area right here that goes through the cell, the plasma membrane of the cell, you'll see that they look like they're little curls. This is called alpha helix. They actually twirl around like this so that they can give themselves more stability and more structure inside. Okay? So this one has 7 transmembrane domains. So it takes up a pretty good piece of the membrane, okay? You also see that it has this big loop on the outside, and it has a big loop on the inside. So all the G proteins are going to have the N terminus on the outside, extracellular, and the C terminus on the inside, which is the intracellular. Now, this transmembrane protein is the receptor, but it's not actually the G protein. The G protein is coupled to it. And actually, if you count down here, 1, 2, 3, the alpha subunit of the G protein is going to connect right there to this loop on the inside. And then the beta gamma subunits are going to connect right here to this green section. Okay? Now, one of the ways that we make a whole bunch of different ones of these is we make a whole bunch of different ends. You can see how this one looks like it's covering up the top here. And we make a whole bunch of C terminals down here so that we can hook other proteins to them. Okay? So that's how we get the variation is mostly in the ends. Because what's inside here is going to be pretty conserved. It's going to be pretty much the same for all of them. Okay? But that's what this picture is trying to explain to you, is how it looks and how it does its job. Okay? So when you look at this one, you're going to notice that the G protein isn't shown attached to the transmembrane. Okay? That is simply to make it easier for you to see it. But in reality, it's always going to be attached right there on that third ring, on that third loop right there. So you can just imagine when you look at the pictures that it's actually attached. Okay? So what's going to happen is this 3 subunit G protein right here is going to be activated by the ligand binding to the receptor. And up here at the top, there's a whole list of different ligands that can possibly bind to that, okay? Just to give you some ideas of the variety, okay? So what's going to happen is that it's going to activate the alpha subunit, which can be alpha S stimulatory, alpha I inhibitory, or alpha Q, and actually there's even a few more new ones after that. And that is going to separate. So the B, the beta, and the gamma are going to stay close to the membrane, okay? But the alpha is not. The alpha is going to go into the cytosol, okay? And you'll see that the way that it does that is with that GTP, okay? That's where the activation comes from, okay? And if you look down here, it gives you a whole list of possibilities of things those alpha subunits can do, okay? So one of them increases cyclic AMP and one of them decreases cyclic AMP. There's the balance, okay? Remember, we said stimulatory and inhibitory, okay? You'll also notice that this part just goes along and it has a whole other function, and its function is to go activate a different signaling mechanism. Okay, so we could say that the alpha subunit is going to activate here, but this one's going to activate over here, and that PLC stands for phospholipokinase. No. It just blew out of my mind. It'll come back. I'll figure it out in a minute.